So synthetic mRNAs came to the fore during the COVID-19 pandemic, which showed that they're both safe and effective and can be manufactured at a massive scale. But since then, there's been significant investment in expanding the utility of both mRNA vaccines and further therapeutics. So in addition to developing more and more mRNA vaccines, which was really nicely described by Andreas from BioNTech this morning, there's also a lot of protein replacement therapies that are being developed to treat rare diseases, and also somatic sequencing-related personalized cancer medicines that are being developed. In addition to this, synthetic RNAs can also be used for genome editing, gene knockout, gene knock-in, and epigenetic-based editing. So you can see there's a huge potential for our new mRNA therapeutics. So I come from the base facility at the University of Queensland. We're Australia's largest manufacturer of research-grade mRNAs. This year, I think we've prepared more than 300 mRNAs to date. So we're really helping to support Australian mRNA research. And we're also establishing clinical-grade manufacturing. So BASE provides an end-to-end service that includes mRNA design, manufacture, formulation, and validation. So I guess this has already been covered pretty well already, but just quickly, mRNA vaccines mimic a natural mRNA. They have a 5' cap, 3' and 5' UTRs that stabilize the transcript and enable translation, and a coding region that could really include anything which was mentioned earlier, antibodies, protein replacement therapies, etc., and also a poly-A tail. Now, the exact sequence of these mRNA vaccines is very important for controlling the efficacy and the stability of these mRNAs. But how do we make these mRNAs? Well, first of all, you start with a plasmid template. The plasmid template encodes the full mRNA sequence. It's then linearized, 3' to the poly-A tail, and then it's used as a template for an in vitro transcription reaction. Now, that mRNA is then purified and formulated and can then be subject to final product characterization. Now, really, what's important is that during the in vitro transcription reaction, anything can go wrong, right? You can have SNPs, for example, that might get introduced. There could be truncations. You could have transcription that ends before the end of the mRNA. And also, during the plasmid propagation step, there can be truncations in the plasmid template. And all of these can have major influences on the efficacy and safety of these mRNAs. So understanding whether or not they've arisen during manufacturing is extremely important. So this has led the USP to come up with a set of guidelines for characterizing mRNA vaccines. Again, this has been covered really well. This is only a subset of what's asked for, but you can see it's fairly intensive. It's very time-consuming to do all of this testing. And we thought, why not try Oxford nanopore sequencing to try and streamline this testing that requires a larger number of devices and a very long period of time? So we did just that. In 2023, we published a Nature Communications paper where we outlined a proof of principle that nanopore sequencing could be used throughout mRNA vaccine manufacturing. And then it caught the attention of Oxford Nanopore, who then entered into a research partnership with us to try and improve this test and potentially to bring it to market. So it led to the development of this mRNA identity QC test, which I'll explain today. So what we were really trying to do was to look at how Oxford Nanopore sequencing could be used throughout mRNA vaccine manufacturing, in the validation of plasmid templates, in drug substance testing, and also in final product testing. So first of all, plasmid release testing. So this is work that was done by my colleague Sneha Lakshman-Das, who generated a whole lot of excellent data. So what she did was she generated some rapid sequencing libraries from 4 different mRNA vaccine templates and then sequenced them for 72 hours. And you can see right off the bat here, she's getting in the multi-million reads within the 72 hours, which was really a lot. And yeah, so that's like more data than you would ever want. So what we wanted to do was try and understand whether maybe we could subsample that a little bit. So as a first port of call, we wanted to look at the way that error profiles change over the course of the run. Can we capture just a subset of the sequence that might be a little bit more reliable? Now, what you can see here is that there is actually a correlation between sequencing time and mismatch and deletion errors. So you're getting an accumulation of more mismatch and deletion errors as you go further into the run. So from this, we reasoned that capturing the first part of the sequencing, first 200,000 reads, should be a way of improving the overall quality of the data. And so this is what it looks like. If you take these hundreds of thousands of reads and you align them against the reference genome, the reference sequence, then you can see that it covers the full plasmid. What you can see here is you can see basically where the plasmid has been linearized at the end of the poly A tail. And that allows us to investigate, but by looking at read start site, it allows us to investigate whether or not some of the plasmids aren't quite linearized. We can confirm the sequence identity. And also what we were able to do is try mapping these reads against the E. coli genome. And that allowed us to work out whether or not we had any E. coli DNA that was being carried through the MaxiPrep. So what's also really interesting, we wanted to know how sensitively we might be able to pick up any subclonal variation that might have arisen during plasmid propagation. So we did some subclonal variant testing. We designed 4 different COVID-19 mRNAs that were encoded into plasmids from 4 different spike protein variants. And we found that they had about 85% similarity when we compared them. And we combined these at different concentrations, between 90%, 10%, 1%, and 0.1%. We spiked them together and then sequenced them. And we wanted to know for each individual nucleotide that was sort of private alleles for these different mRNAs, whether we could detect them above the background sequencing error rate. And we found that the plasmids that were spiked in at 1% or more of the total pool could be detected reliably above the background error rate. So that's quite cool. So it's 1% sensitivity to detect subclonal variants based on our analysis. And what's also really important is that we got consistent, this result was consistent at both 2 hours and 72 hours. So there was absolutely no gain in sensitivity by sequencing for a further 70 hours. So next, mRNA drug substance testing. So after we've used this plasmid template, it's been release tested, and we've even in vitro transcribed it and cleaned it up, how can we analyze it? So this is, again, work that was done in collaboration with Sneha Lakshman Das and Magdalena Budzinska, who designed a next flow pipeline that we used for this research. So Sneha prepared RNA-004 libraries with no reverse transcription or PCR step, as you know. They were base called in Dorado, and this base calling used a custom N1-methylpseudouridine base caller. Now, why would we want to do that? Well, it's very common to incorporate modified bases into mRNA vaccines, and this is why. If you use unmodified nucleosides in your mRNA vaccine, you're going to have a really high level of inflammatory response, and that's going to basically kill your RNA and massively reduce the amount of translation of your protein. However, if you use modified bases, such as N1-methylpseudouridine, which is used in the COVID mRNA vaccines, then you have a massive increase in your translation rates, also a reduction in the inflammatory response. And so what's really cool is that by using this N1-methylpseudouridine-aware base caller, we're able to use RNA-004 sequencing, which sequences the native mRNA without any cDNA, basically. So this is incorporated into our pipeline. So after doing the base calling, we then aligned the reads to using Minimap2, and then analyzed the accuracy using BCF tools. And we were able to produce a QC report that was pretty exhaustive for a range of different quality attributes. So how did we test and validate this method? Similarly to the plasmids, we prepared mRNAs based on the same plasmid templates that were described before, and in addition to Cas9. And then we made these mRNAs with either N1-methylpseudouridine or canonical uridine. And again, we got more than 1 million reads, and this time within 24 hours. So again, more reads than you could ever dream of, basically. So again, we subsampled them to 200,000 reads to put it into more, which is again less than 2 hours, to put it into a more realistic time frame for testing during manufacture. So this is what the data look like. What you can see here, again, the mRNA reads are aligned against the plasmid reference. And the vast majority of the reads, you can see here, that basically these grey bars at the top here are mostly aligning against the mRNA coding region. You do see a small number of reads that are aligning on the plasmid backbone. Some of them are the complete plasmid backbone, which are probably the product of incomplete linearization of the plasmids, or also transcription that was initiated spuriously against the template and not against the T7 promoter that's incorporated into the plasmid template. So what do the error rates look like? Well, pretty good. when we align against the reference, excluding the poly A tail, the median sequencing accuracy was about 91 to 95%. I think it's important to say here now that there's been a revised base caller, which has brought that accuracy right up as well. So it's a shifting and improving target there. Despite the fact that we excluded the poly A tail from the reference, deletion errors were still the most abundant error type for our different constructs, followed by mismatch, and then we had almost no insertion errors. Next, we were able to look at the sequence identity. So for all of our tested templates, we were able to show 100% sequence identity in comparison to the reference. And this is important because we want to be able to pick up whether or not there's been any rearrangements or translocations or perhaps any SNPs that have accumulated during the manufacture. So the report that we've been working on generates dot plots and also consensus alignments. We were also able to look at integrity. So something that's important about long read sequencing, which I probably don't need to tell you, is that that if you used short read sequencing, you wouldn't be able to work out anything to do with the integrity of the mRNA. If you're using inherently short chunks of sequence, then you're not going to see how many fully intact mRNAs there are. Whereas with nanopore sequencing, we were able to sequence the full mRNAs and look at putatively how many functional mRNAs there were in the pool. Again, we were able to use the mRNA coverage as a proxy for integrity for each of them. Yeah. So something that's important to note as well, even though we were approaching 100% coverage for all of our different constructs, some of you probably know that the extreme 5' end of the mRNAs gets a little bit cut off, about the first 10 nucleotides. When you're doing direct RNA sequencing, and that's the nature of the sequencing. You don't have a 5' adapter, so it tends to go through the pore quite rapidly at that point. And so those are omitted currently. So that's a very slight limitation of that platform. We were also able to look at poly A tail length. Now these are plots that we generated. Basically what you can do is you can take the sequencing output from Dorado and you can identify whether or not truncations might have happened during the mRNA manufacture. What you can see here, these are mRNAs that were transcribed from plasmid templates. The majority of the mRNAs had a fully intact poly A tail at around 120 nucleotides. However, you can see quite a few transcripts that were about 16, that had poly A tails that were around 60 nucleotides. We believe this is because of truncation of the poly A tail that happened during the plasmid propagation step. It's results like this that's led base to adopt a different type of template. We now use a combination of a DNA template and PCR, which is amplified through PCR, and it's associated with a much more intact poly A tail, which is pretty cool. So this information has been really extremely useful for our own R&D. Another cool thing that we can do is we can identify the 5' cap of the poly A tail. At the moment, we use cDNA sequencing to do this. How do we do it? Well, we're using CleanCap AG, so co-transcriptional capping, during the mRNA manufacturing, and we've noticed that that during the transcription of the mRNA, you have an additional nucleotide on the 5' end of a capped mRNA. So the transcription initiation site changes subtly from a G to an A, depending on whether or not you have incorporated a cap. And so just by looking at the proportion of reads that have that additional G at the 5' end, we can there's a linear relationship to the abundance of the caps that are in the mix, which is pretty cool. So how can we use it in final product characterization? So the mRNA has been in vitro transcribed, cleaned up. We're super happy with it, and now we put it into LNPs. How can we analyze using nanopore? Well, so as I said, LNPs during the encapsulation process, the mRNAs are exposed to mechanical forces and heat that could potentially lead to degradation of the mRNAs. And so it's really important to work out whether or not the integrity has changed. So we developed a method that strips the mRNAs from their LNPs. We got super high quality, high purity mRNAs by doing this. I can discuss the protocol with you afterwards if you're interested. What we were able to do was to compare mRNAs that hadn't been encapsulated to those that have been removed from their encapsulation. And I hope you can see here that it had very limited impact on the total read length. You see a very slight increase in the number of degraded reads after formulation. But this was pretty informative, for our own R&D efforts in working out whether or not our LNP encapsulation was impacting mRNA integrity. Another cool and pretty unique thing that you can do with final characterization using nanopore is to analyze multivalent mRNA vaccines. So if you were to Sanger sequence a multivalent mRNA vaccine that's incorporating a range of different variants or different templates, then you're going to get a mess. But long-read sequencing, of course, allows you to look on the single molecule level and identify different mRNAs. So what we did was we made a mock multivalent vaccine using EGFP, mCherry, ovalbumin, and luciferase. We spiked them together at 4 different concentrations between 10 and 40%. And then used them, again, without any kind of barcoding. We made RNA-004 libraries with them. What we were able to do is to show not only could we uniquely map all of our different single molecule reads against their reference sequences, the count of those reads matched the expected proportion in our mix. So it was good for not just confirming the sequences and the integrity of these individual reads. We could also look at the relative abundance and whether or not we'd pooled them correctly. So in summary, we were able to test whether nanopore sequencing could be used throughout mRNA manufacture, from plasmid DNA release testing to drug substance testing and to drug product testing. And we were able to demonstrate that a lot of these different potential applications worked out. That indeed, we can use mRNA, Oxford nanopore sequencing throughout mRNA manufacture. And there are still some elements of it that are in development. For example, the 5' capping and the double-stranded RNA is still under development to have a single united RNA-004-based test. But watch this space, basically. So just in the last 2 minutes, I'll talk to you about how BASE is adapting this test for use in clinical manufacturing. So Colette really did a great job of talking about why the Q line is a great idea for developing a GMP grade test. So we're moving towards clinical RNA manufacturing, and we would like to be able to use the GridION Q as part of our validation procedures for the reasons that Colette went through. That basically, if you're going to be validating a protocol and writing strict SOPs, you want to have a locked down product that isn't going to be hit with fantastic new updates all the time. And so we wanted to look at whether this could form part of the final product characterization amongst other methods. Now, while we think the GridION Q is great and it can fit amongst other testing, it's not going to do everything ever. Things like mRNA concentration and purity, pH and conductivity, residual pDNA are unlikely to ever really get taken on by Oxford nanopore sequencing. So you can see here it would be one of a suite of different techniques that can be applied to final product characterization. And one of the challenges in incorporating that in a range of different testing modalities is to do AIQ to work out whether or not this device is really suitable for testing this range of different parameters. So to do that, we've developed some different mRNA standards, and these, again, these can be used for different types of validation throughout manufacturing. First of all, in initial device qualification, so the OQ, PQ, and then in requalification after repairs and maintenance, etc. And these are designed to look at the integrity of the mRNA, the inclusion of double-stranded RNAs, the efficiency of 5' capping, different poly-A tail lengths, and types of fragmentation. So these are being manufactured as we speak at the base facility, and we do plan to make them available to the community. So if you're interested in these, then get in touch after the session. Yeah, and the idea is that these will be really super useful in indexing against user acceptance criteria and specifications. So in summary, I don't know if any of you like Bluey, but yeah, Bluey is made basically in where I come from. And so basically what we've been able to do is to use Oxford nanopore sequencing for a cap-to-tail analysis of mRNA vaccines. They're suitable for use throughout mRNA manufacturing, including final product characterization. In particular, and interestingly, this includes multivalent vaccines, and also this mRNA quality test is being adapted for use in manufacture, but we're in much earlier stages and still watch this space. So I just wanted to acknowledge my colleagues at the base facility, especially Sneha Lakshmandas and Magdalena Budzinska, and also my collaborators from Nanopore in helping to bring this together. Thank you so much.